Hi, this is Sean Kim and Jonathan Lerner, and today we're going to be discussing the article Engineered Spider Silk-Based 2D and 3D Materials Prevent Microbial Infestations. Why is this an important issue? Well, often biofilms formed from microbes that are immune to antibiotics like fungi and bacteria, and these biofilms can harm patients by lowering the effectiveness of biomedical devices. And as a preventative measure, spider silk works because it has mechanical properties like strength that surpasses other materials. It is biocompatible with biomedical applications, and it resists and repels microbes to a very effective degree. And these spider silk proteins were used to create 2D to 3D scaffolds that resist microbial decomposition. And often, recombinant DNA technology studies the innate anti-fouling capacity of spider silk, which resist attachment to surfaces, and it uses spider silk proteins to create biomedical devices like films and hydrogels.
Film formation on 2D surfaces using E. coli and P. pastoris was first tested on 4 variants of spider silk. One spider silk-based material that varies in amino acid sequences or protein structure, and a PCL film, which is just a standard film. Spider silk performs substantially better with lower bacteria adhesion rate and adhesion force after washing the surfaces of each material tested to ensure that the bacteria was truly adhered to the surface. To ensure this wasn't a topographical feature, surface feature of the silk, another experiment was performed where each material had grooves of different sizes placed in it, and it yielded the same result as the previous experiment.
Another experiment was done this time with hydrogels. 3 hydrogels containing spider silk proteins and 2 control hydrogels were incubated with E. coli and P. pastoris at 37 degrees Celsius for 24 hours. After which, all hydrogel samples were washed to remove non-adhering bacteria and microbes, and an Almer blue viability assay was used to determine how much E. coli and P. pastoris adhered to the surfaces and survived. And as seen from the graphs here, the 3 spider silk protein hydrogels, the 3 on the left, had little of the E. coli and P. pastoris left, while the other 2 control hydrogels had vast amounts of microbes remaining. So this shows that spider silk protein is very effective at repelling microbes.
Researchers conducted a test on a variant of spider silk which is known to promote mammalian cell adhesion to see if spider silk can be used to promote bacteria-free tissue growth, like on a cut. Hydrogels encapsulated animal tissue, which were inoculated or had bacteria placed in it, and were allowed to incubate for 6 hours. The samples were washed thoroughly afterward to remove bacteria that was not adhered to the hydrogel. There was no measured adhesion of bacteria in either of the hydrogels, the variant or the original, but the spider silk variant did stimulate cell growth as expected.
So as we see here, recombinant spider silk materials and spider silk proteins have the capacity to repel microbes and stop biofilms from forming, often due to little hydrophobic space present to let microbes form. And then modified spider silk with homogeneous hydrophobic spaces will be capable of bioselectivity, where they allow mammalian cells to adhere and grow while causing microbes to repel away. And this, interestingly, only the fundamental DNA structure of spider silk proteins are needed to maintain the spider silk microbe repellent surface. So a lot of opportunities are available for spider silk material in the biomedical field.
Adhesion force measurements were conducted by taking a force distance measurement using the bioscope catalyst, a microscope that measures atomic force as seen in the top right. Microbial adhesion was measured by taking the surface of the hydrogel, placing it in a fresh media, and culturing for another 12 hours at 37 degrees Celsius, which was measured by the OD600 dilute meter, a machine designed to measure cell growth, which can be seen on the middle right.